The Kalahari Desert stretches across roughly 1 million square kilometers of southern Africa, covering parts of Botswana, Namibia, South Africa, and Zimbabwe. It is technically a desert in the way the Sahara is a desert. It receives more rainfall, it supports more vegetation, it hides beneath its red-orange sand a surprisingly complex ecosystem of grasses and acacia trees and wildlife that have adapted across millions of years to this semi-arid landscape. But it is dry, it is vast, and it is remote in a way that has meant the people who have lived here for the longest time have done so largely without the notice of the civilizations that rose and fell in other parts of the world. The Sand People, sometimes called the Bushmen, a name they use themselves, though it carries colonial weight, have been living in this landscape for longer than any other identifiable human group has been living anywhere. Not longer by a few centuries, not longer by a few millennia, longer by a span of time that, when you write it out as a number, changes the shape of the question of who we are and where we come, for more completely than anything else that modern genetics has discovered. Somewhere in the region of 90,000 years ago, before the ancestors of every non-African person alive today had left the continent, before any farming had been invented anywhere on Earth, before any city or monument or writing system existed anywhere in the world, before the Neanderthals, who would later interbreed without migrating Homo sapiens, had even reached the peak of their range in Europe. The genetic lineage of the San had already diverged from the lineage that would eventually produce all other modern humans. The San were already a distinct, separated population before the world that produced everyone else had even begun. This is what modern genetics found when scientists compared the DNA of populations from every corner of the Earth and asked, for the first time, systematically and with sufficient data to answer properly, which human populations are oldest, which lineages diverged earliest from the common ancestor of all living humans, whose genome has been evolving in place, in continuity with the most ancient human populations, for the longest unbroken span of time. The question sounds simple. The answer sounds like it should have been obvious. And the reason it shocked the scientific community and has continued to generate awe in everyone who examines the data carefully is not just because of what it found. It is because of what it found in combination with what most of human history has believed about the relationship between these specific populations and the concept of progress, civilization, and human worth. Everything that has ever been said about human racial hierarchy, every classification system that placed some populations above the others in intelligence and development and biological sophistication, was aimed in varying degrees of explicitness at claiming that the populations labeled primitive or a savage by European colonial ideology were somehow less advanced, less developed, less fully human than the populations producing that ideology. The San, the Hadza, the forest peoples of Central Africa, the hunter-gatherers who maintained the oldest ways of human living, were placed at the bottom of every hierarchy that European colonialism constructed. They were the ones colonial science most aggressively declared were closer to something pre-human. And they are the ones who carry the oldest human DNA on Earth. Not older in some loose, figurative sense. Older in the specific, precise, technically rigorous sense that the deepest rooting branches of the human mitochondrial family tree, the genetic record of maternal lineages across the entirety of human history, belong to these populations and to no others. The ranking of human populations, by the age of their DNA lineages, produces a result that is the exact inverse of every racial hierarchy ever proposed. And understanding why that result is meaningful and what it tells us about what oldest actually means in genetics requires understanding the tools that produced it. Mitochondrial DNA is the key. Unlike the nuclear genome, the 3 billion letter sequence in the nucleus of each cell, mitochondrial DNA is a small circular molecule, only about 16 out of 500 base pairs long, that sits in the mitochondria, the energy-producing structures that float in the cytoplasm outside the cell nucleus. Mitochondrial DNA has one unusual property that makes it extraordinarily useful for tracing human evolutionary history. It is passed from mother to child without mixing with the father's DNA. A son inherits his mitochondrial DNA from his mother, but he does not pass it to his children. A daughter inherits it from her mother and passes it to her children. Every mitochondrial lineage traces in a straight line through maternal ancestry, generation after generation, without shuffling. Over time, mutations accumulate in mitochondrial DNA at a roughly measurable rate. Each mutation that occurs in a lineage and gets passed on to all subsequent generations is a time marker, a molecular clock tick that allows researchers to calculate approximately how long ago two lineages shared a common ancestor. By comparing the mitochondrial DNA of living people from populations around the world and mapping the pattern of mutations that separate different lineages from each other, geneticists can construct a tree, technically called a phylogenetic tree, that shows how all living human maternal lineages are related to each other and, approximately, when they diverged. When researchers built this tree using samples from populations across the globe, they found that all living human mitochondrial lineages trace back to a single ancestral lineage, called mitochondrial haplogroup L, whose origin is estimated at approximately 145,000 to 200,000 years ago in Africa. This common ancestral point is what popular science calls mitochondrial Eve. Though the name is misleading, it does not mean there was only one woman alive at the Timii whose descendants are all of us. It means that of all the women alive at that time, only one has a lineage that has survived D to the present in an unbroken maternal chain across all subsequent generations. All other maternal lineages from that era eventually died out when their carriers had no daughters. The tree that branches out from this ancestral lineage has deep roots in southern Africa. The first branching events, the earliest splits within the human mitochondrial tree, produced lineages labeled L0, the oldest of the L haplogroups, whose earliest diverging subbranches are L0D and L0K. And these haplogroups are found today primarily in one place, among the San and Khoisan-speaking populations of southern Africa. The numbers attached to these haplogroups are not approximations. They are peer-reviewed estimates from multiple independent studies using different methodologies, producing consistent results. L0D, the earliest diverging maternal human lineage currently known to exist in living people, has a coalescence time estimated at approximately 172,000 years, with a 95% confidence interval of 149,000 to 199,000 years. L0K, the next earliest diverging lineage, has a coalescence time of approximately 159,000 years. The broader split between the Khoisan lineage as a whole and all other modern human lineages has been dated to approximately 90,000 to 100,000 years ago. These specific haplogroups, L0D and L0K, are most common precisely among the San people of southern Africa and the Khoisan-speaking populations of Namibia and Botswana. To put the 172,000-year date in context, the out-of-Africa migration that populated the rest of the world with the ancestors of every non-African person alive today occurred approximately 50,000 to 70,000 years ago. By the time that migration happened, by the time the populations that would eventually become European, Asian, Polynesian, and indigenous American had even left the continent where humanity began, the L0D lineage of the San had already been a separate, distinct genetic lineage for at least 100,000 years. The San lineage was ancient when the ancestors of every other population on Earth were still in Africa. It has continued in that same place for all the time since. The Y-chromosome tells a parallel story. Like mitochondrial DNA, the Y-chromosome, which determines biological maleness and is passed from father to son without recombination, traces paternal lineages across time through the accumulation of mutations that serve as molecular timestamps. The oldest known human Y-chromosome lineage is haplogroup A00, discovered in 2013 by researchers who found it in a population called the Mbo from Cameroon in central West Africa, and independently confirmed in African American men whose ancestry traced to the same region. The estimated age of haplogroup A00 is extraordinary, approximately 340,000 years, well before the earliest dates assigned to anatomically modern Homo sapiens in the fossil record, and predating the emergence of our species as conventionally defined. The haplogroup's discovery prompted significant scientific discussion about what it implies for the origin and early population structure of modern humans, with some researchers suggesting it may reflect interbreeding between early modern humans and a more archaic population in Central Africa, during a period for which the fossil record is sparse. The specific populations that carry these oldest lineages form a coherent geographic and cultural group: the San and Khoisan speakers of southern Africa, the Hadza of Tanzania, who carry ancient L0 haplogroup variants and show evidence of shared ancestry with Khoisan populations, the Sandawe of Tanzania, who similarly carry deep L0 lineages, the forest-dwelling pygmy populations of Central Africa, who also rank among the earliest branching lineages on both the maternal and paternal trees, Sarah Tishkoff of the University of Pennsylvania, who has spent her career on African genomic diversity and who led one of the most comprehensive studies ever conducted on the subject, found in her 2009 science study that the San of southern Africa were among the populations with the most diverse nuclear DNA of any group on Earth, confirming in autosomal sequence data what the mitochondrial and Y-chromosome analyses had shown in uniparental lineages. The diversity finding is inseparable from the age finding, and understanding why requires understanding what diversity means in this context. All non-African human populations are genetically less diverse than African populations. Not less diverse in every individual comparison, but less diverse on average, and and less diverse in a systematic and directional way that reflects a specific historical event. When the ancestors of all non-African humans left Africa approximately 50,000 to 70,000 years ago, they were a subset, a relatively small group of people who carried a subset of the genetic variation present in the African populations they left behind. Every non-African population today descends from that small founding group, and the genetic variation present in non-African populations reflects only the variation that was present in that founding group. All the additional variation that existed in the African populations who did not migrate, all the additional lineages, all the additional mutations, all the additional diversity that had been accumulating in Africa for tens of thousands of years before the migration remained in Africa. Tishkoff's 2009 study found that Africa contained more genetic diversity than the rest of the world combined. The ancestry found outside Africa is, as she put it, largely a subset of the ancestry within it. This is the genetic reality that the racial hierarchy inverted so completely and so consequentially. The populations labeled primitive, the hunter-gatherers of Africa who had maintained the oldest continuous human way of life, were labeled as such precisely because they had not participated in the transitions to agriculture, to cities, to writing, to the forms of material complexity that European colonialism used as its benchmarks of human development. But genetic age is not a measure of cultural complexity. It is a measure of how long a lineage has been in continuous existence in its current geographic location, accumulating diversity, surviving through climate shifts and demographic pressures, maintaining its biological continuity. The populations that did not migrate out of Africa, that did not go through the genetic bottleneck of the out-of-Africa dispersal, that remained where the human lineage began, those populations are genetically oldest precisely because they stayed. The sand did not lag behind the rest of humanity. They were the rest of humanity's point of origin, and they remained at that point of origin, while everyone else spread out across a world that the sand's ancestors had been inhabiting for tens of thousands of years before the migration began. The concept of ranking populations by DNA age requires one more careful clarification, because the headline framing of that question can imply something that the genetics does not support. Saying that the sand carry the oldest DNA lineages does not mean that individual sand people are biologically older than other people. Every living human being has ancestors that extend back the same number of generations to the same common ancestors. Every living human being is equally modern, equally evolved, equally Homo sapiens. What differs between populations is not the individual's biological age, but the age of the lineage that distinguishes them from other populations, the specific genetic branch of the human family tree that their specific ancestry represents. The Sands branch is longer in the specific sense that their distinguishing lineage diverged from the common trunk earlier than any other living population's distinguishing lineage. This is a statement about population history, not about individual biology or intelligence or cultural value, or any of the qualities that racial hierarchies attempted to rank. The direction of the ranking, though, is what makes the genetics politically and historically significant in ways that extend beyond the purely scientific. The hierarchy that colonialism built was anchored in a claim about which populations were most developed, most advanced, most cognitively capable. Modern genetics has provided a hierarchy based on a completely different and more objective criterion, the age of genetic lineages. And that hierarchy ranks the populations that colonialism placed at the bottom at the very top, and ranks the populations that colonialism placed at the top, closer to the bottom, as genetically younger, as descended from a subset of the diversity carried by the populations that stayed in Africa while the rest of the human story was spreading outward across the globe. The Hadza of Tanzania, who have maintained a hunter-gatherer existence in the region of Lake Eyasi for tens of thousands of years, and who carry ancient L0 lineages related to the Khoisan populations of southern Africa, numbered approximately 1,000 to 1,300 people, according to the most recent census. The San of the Kalahari, whose lineage has been continuous in southern Africa for longer than any other identifiable human population anywhere on Earth, face ongoing pressure owing in their land rights, their access to the territories they have occupied since the deepest reaches of recorded human genetic history, and the cultural continuity of a way of life that represents the longest surviving human tradition on the planet. The populations that carry humanity's deepest roots are also, by most measures of contemporary material circumstance, among the most marginalized. That is the complete picture that the genetics reveals when scientists rank human populations by the age of their DNA. Not a comfortable picture, not a simple one, a layered one, in which the biological finding about lineage age intersects with a historical record of what has been done to the populations who carry those lineages in ways that the genetic data does not resolve, but that the genetic data makes impossible to ignore. The San carry the oldest maternal lineages in the human species. The Embo of Cameroon carry the oldest known paternal lineage. The hunter-gatherers of Africa, taken together, represent the deepest roots of human genetic diversity, the populations closest to the common ancestors of all living humans. This is not a metaphor. It is a measurement made with sequencing technology, validated across multiple independent studies, published in peer-reviewed journals, and reproducible by any laboratory with access to the appropriate samples. The oldest humans on Earth are not who history taught anyone to expect. And understanding why they are who they are is essential to understanding what we all are and where all of us came from. Subscribe, because the people who carry the oldest story on Earth deserve to have that story told accurately.